as we take antibiotics out of prophylactic antibiotics or growth promoting antibiotics out of the feed, we recognize that allows for more variation in the microbiome and more sudden changes in the microbiome. And often it's not the fact that a particular bacterial species is present or not present in the gut. It's when are the conditions favorable for that particular species to proliferate and grow and start causing problems. So, it's really brought into focus how important the gut is as an immune organ. And if we think about it, it's really the largest point of contact with the outside environment.
Hello and welcome. I'm Kate Milosh, your host for today's episode of the Poultry Podcast Show. Joining us is our guest, Dr. Doug Korver. Dr. Korver completed his undergraduate degree at the University of Saskatchewan. He went on to complete his master's at the University of Delaware and his PhD from the University of California, Davis. He is currently a professor of poultry nutrition in the Agricultural, Food, and Nutrition Science Department at the University of Alberta in Edmonton. In addition to practical research on feedstuff quality and the evaluation of dietary supplements in poultry diets, Dr. Korver's work focuses on interactions between nutrition and immune function, as well as bone development in both broilers and layers. Dr. Korver also teaches animal nutrition courses at undergraduate and graduate levels, ranging from introduction to animal nutrition all the way up to advanced nutrition and metabolism. He has authored or co-authored over 75 peer-reviewed articles and 14 book chapters. In 2021, he was named a Fellow of the Poultry Science Association. He is currently part of the National Academies of Science, Engineering, and Medicine's Committee to revise and update the 1994 nutrient requirements of poultry. Thank you so much for your time, and welcome to the show, Dr. Korver.
Thank you, Kate. I'm happy to be here.
How are you doing today?
Very good.
Good. Well, before we begin talking a little bit about your research, could you tell us the story of how you came to be a professor at the University of Alberta?
Sure. I've always had an interest in animals and in particular birds. And so as a high school student and even before, I had an eye on a veterinary career. I went to university with that in mind. And let's just say my first couple of years were not stellar academically. And so that kind of took me out of the running for being competitive. But as I started to figure things out academically, I also had a chance to work for the poultry unit at the University of Saskatchewan. And so I really had an opportunity to get a lot more hands-on experience working with poultry. I had an opportunity to do a small research project as an undergrad, and I really enjoyed that. And so the career goals changed. I decided that I'd look at doing a master's degree in poultry nutrition, which I did at the University of Delaware. And I really enjoyed that. And then I had an opportunity to do a PhD in California with Kirk Klasing. And so by that time, I was really invested in it. I really enjoyed it. And I saw lots of opportunities for the future. And so I wouldn't say I set out to become a professor, but certainly the doors opened up along the way, and it's been a fun ride.
Mm-hmm. I would say that pre-vet transition into more poultry industry, poultry nutrition is a pretty common career path that we hear a lot.
I think so, yes.
Yeah. A lot of us grow up loving animals and don't really know anything else as a career other than being a veterinarian, and there's so much more out there.
Well, and I see that a lot in the students that I teach as well, is they come in thinking they want to be vets, and then as they get exposed to more agriculture in general, and in my field in poultry particularly, they realize that there's a lot of interesting things that they can do that, even with a veterinary degree, they wouldn't be able to do, or there's opportunities that they wouldn't necessarily have expected coming into it.
Absolutely. So throughout your career, was there any time that you can think of where you thought, why am I doing this? And if so, what made you keep going?
Usually around grant writing season. So, it's always an interesting process, but, it usually, the grant deadlines are never at a convenient time. And so you're always trying to juggle a few different things all at once. So I wouldn't say that that's something that, I question, why am I doing this? It's part of the job. And it's just one of those things that you have to get through. And then, hopefully when your grants are successful, then you can do the fun stuff. So
yeah, it's a long wait for the payoff on those too. I think that makes it even harder, that sometimes it takes a while to hear back.
exactly.
Yeah. If you can think back over the last 10 or so years, is there anything that you know now that you wish you had known then?
Yeah, that's a great question. And maybe it's not even just the last 10 years, but one of the things that I would encourage anybody in the poultry field is to look for opportunities to get to know people, to collaborate. Collaborations outside of your institution have really, in my case, have really picked up over the last 10 years. In large part, it's because I think it is a fairly small community, the poultry nutrition community. We feed billions of birds around the world, but there's a fairly small group of people that are doing the kinds of work that we do. And so scientific meetings and industry meetings are a great chance to get to know people. And just through chance meetings in hallways, you strike up a conversation and you realize that there's a common interest. And so that's an encouragement that I would give to people starting in their careers. Look for those external collaborations. Great to collaborate within an institution and important, and those are the people you're going to work with day to day. But there's a world of opportunities if you're interacting with people across the country and across the world.
Mm-hmm. Definitely looking at the history of your publications and all of the presentations and conferences that you've been involved with. I mean, you must have made so many different connections with people across the industry, and that's so important.
Yeah. And
looking forward, is there anything on the horizon that you're excited about career-wise?
Yeah. So one of the things that's happened over the years is as I've developed more of these connections, I've been invited to more and more conferences, giving industry talks. And so I'm about to embark in a bit of a change in my career. So I'm cutting back my time at the University of Alberta. I'll still be there part-time, but I'm building up a consulting business. And so I'll be looking to interact with ingredient suppliers and poultry companies, primarily looking at, I still see myself as a teacher. So a lot of what I want to do is taking the technical and scientific information that's available and translating that to the end user. So putting it in context, gathering information from broad sources, and trying to focus on what the end user will need to know in order to make their operation successful.
That's so exciting. Congratulations on that upcoming change. Of course, everyone is glad to have had you at the university, but also for you to be able to get out and share that knowledge across the industry and with folks on the feed additive side. It's definitely always good to have, a broader range of knowledge applied across all of these areas. I know people will be very fortunate to have your help in that regard.
Yeah, and I'm really looking forward to it because I'll still have a relationship with the University of Alberta, so I'll still do a little bit of teaching at the undergraduate level. And I'll have my research program, so I'll still be generating new knowledge and hopefully staying relevant to the industry in that way as well.
That's great. What a great way to bridge that gap between industry and academia to kind of, have connections in both.
I'm looking forward to it.
Yeah, sounds really fun. So as far as your research is concerned, what would you say the core mission of your research program is?
If I had to sum it up, I'd say using basic science to answer practical questions for industry.
That's interesting. Basic science gets a little bit of a bad rap in the industry. I'm sure you're aware of that. I think a lot of people maybe misunderstand the value of it as building up to something that can be applied practically, and that we can't have applied and practical solutions for industry without the basic research behind it to hold it up as a foundation.
Yeah, and there's always a continuum. There's basic science from the standpoint of just discovery and hypothesis-driven research, and finding out more about the biology of the birds. And you know as well as I do that those birds are changing all the time. So the things that we think we have a good handle on now might be a little bit different in 10 years. Some of the problems that we're faced with in 10 years are going to be different. And so the more we understand about the biology of the bird, the better off we will be, or the better place we'll be in order to answer those questions more efficiently for industry. So turning that into a practical application. So
that makes sense. That's a good point. Also, the technology that we have available to conduct research is constantly changing. And I think applying some of these more novel, molecular techniques to poultry is really going to give us the answers to a lot of these questions that are, right now so complicated that we can't possibly answer them from just a practical and applied side.
Exactly.
And specifically, I know one of your major areas of interest is the interaction between nutrition and the immune system and the gut microbiome. Talk about complicated.
Yeah. So in that regard, I'm smart enough to recognize my limitations, and it's really been through collaborations with some of the microbiome researchers that we have at the University of Alberta and collaborations elsewhere. And so, when I talk about taking fundamental research and answering basic questions, it doesn't always have to be an individual. And I think, getting back to my conversations about international collaborations is, we can have more than one set of eyes and more than one set of expertise to answer a question, and we can leverage that power of collaborative research. So again, the techniques in microbiome analysis are changing so rapidly. It's hard to keep up if you're not doing it all the time. But over the years, I've developed some really good collaborations with people that have that equipment and have that knowledge and expertise. And so it's been a fruitful collaboration, I think.
Yeah, I can see that looking at some of the publications coming out of your lab and collaborating labs. Could you tell our audience just some kind of basic background information about what you mean by the interaction of the immune system, the gut microbiome? Because I think when we say immune system, the gut is not something that typically comes to mind. You might think about the thymus and the bursa, bone marrow in mammals.
Yeah, and I think that's really the importance of the gut microbiome is really coming into focus, particularly with the removal of growth-promoting antibiotics in many countries. It's already legislated. In many other countries where it's not legislated, the industry is still working towards that for various reasons: consumer pressure and pressure from large purchasers like the fast food restaurants. And so when we remove a tool that has been very effective, and without getting into the discussions about whether we should or shouldn't or what have you, the reality is we're moving away from antibiotics. And as we take antibiotics out of prophylactic antibiotics or growth-promoting antibiotics out of the feed, we recognize that allows for more variation in the microbiome and more sudden changes in the microbiome. And often it's not the fact that a particular bacterial species is present or not present in the gut. It's when are the conditions favorable for that particular species to proliferate and grow and start causing problems. So, it's really brought into focus how important the gut is as an immune organ. And if we think about it, it's really the largest point of contact with the outside environment. And there's some debate whether it's the lungs or the intestinal tract, but it's certainly a major point of contact with the outside environment. And that's what the immune system does in surveillance mode, is it keeps an eye on what are the contacts with the outside environment. And so, yes, the thymus and the bursa and various other lymphoid tissues and cells are important. But one of the major points of contact for disease is, in fact, the gut, and particularly for disease that's going to reduce growth, performance, and efficiency.
Mm-hmm. Makes a lot of sense. So it would follow then that we can impact the immune system through the gut, through nutrition or feed. Can you think of any examples that come to mind of ways in which we impact the immune system through the feed?
Yeah, sure. So there's really, in my mind, a couple of different levels. So the first level is just preventing interaction between potential pathogens and the gut. And without getting into, a list of various approaches and products, if we can prevent pathogens from attaching to the gut wall, the pathogens need to be resident in order to cause problems. And so if we can do things to prevent their attachment, the pathogens, if they're present, will just be washed through with the digested and excreted. If we can, if the pathogens have established a residency in the gut and we can prevent proliferation, that's going to minimize the interaction, the activation of the local immune response and potentially a systemic response. So the way I view things is the first level is just minimize or prevent as much as possible that unnecessary activation of the immune system. And I say unnecessary because often the things that cause an immune response are not necessarily pathogenic. They're just unusual or changes or unexpected by the immune system. And of course, it's important to have a strong immunological response when there are pathogens present. So if we can reduce the presence of pathogens, if we can reduce the unnecessary stimulations of the inflammatory response, we can maintain performance in that way. The second level is actually within the tissues of the bird. So I always tell my students that the lumen of the gut is actually outside of the bird. It's not part of the bird. But when we look at the tissues, there are cases where there's translocation of pathogens into the tissues, into the bloodstream or into the gut tissues, or if we look at joint problems, it's often gut bacteria that have translocated and ended up in the joint. So in that case, then we look at nutrition to modulate the immune response and in one hand, reduce the systemic inflammation that reduces performance, but also have a strong local and targeted response to that particular pathogen. And so again, there's a number of nutrients that we've looked at over the years: omega-3 fatty acids, beta-glucans, trace minerals, and so on, that we can use to nudge the immune system in the direction that we want it to go. And so when we use those two things together, I think that's the most effective. Minimize the stimulation of the immune system that isn't productive or isn't protective for the animal. And then when there is translocation or there is an actual insult, the ability to quickly and rapidly and effectively deal with it so that the nutrients are not being diverted away from growth in order to support an extended period of high immune response.
So it's kind of a Goldilocks situation. You don't want too little immune response and you don't want too much.
Yeah, and so it's like having a mean old dog on your porch. You know, you don't want him chasing down the neighborhood kids all the time, but you want him there to when it's necessary.
I like that. I noticed you said that not just pathogens are stimulating the gut immune response. I was wondering if you could expand on that. I always wonder when we're least cost formulating diets, you know, the dollars are saying let ingredients come in and out of the diet for whatever makes the most sense economically. I have to wonder, do you think that it negatively impacts the bird or causes an immune response to have that bird see an ever-shifting profile of ingredients in the feed, or to be introduced to new ingredients?
Yeah, so the impact of those changing ingredients is often related to the impact that it has on providing substrate to the microbiome. So especially in broiler chickens, we have a short-lived bird. We have a naive digestive tract in terms of the microbiome that really hasn't reached a stable, mature point by the time that these birds are processed, particularly if we're dealing with a smaller bird market. And so what happens is the microbiome, the microbes are still sorting themselves out. The first bacteria species in a particular niche are not necessarily the ones that are going to be most adapted to that niche. And so they might be there first, but then something else comes along that's more suited and they get outcompeted. So it takes a while for a stable, mature microflora to develop. And so when we're constantly changing the ingredients, we run the risk that now we're creating a new environment where different microbes might be better suited for a particular ecological niche in the gut. And so we really don't develop that stable microflora as quickly. And as I said, even in smaller broilers, they probably haven't even reached that point yet, even in the best case scenario. So, it's a combination of different things. So it's creating conditions where a stable microflora, or as stable as possible given the age of the birds, can be achieved as quickly as possible.
Can you tell us a little bit more about maybe the timeline, for example, in broilers, of how the microflora develops from, the newly hatched chick up to at what point is it typically a stable microflora?
That's a great question.
It probably depends on a lot of factors.
Yeah, it does. And so, one of the things, without putting a specific timeline on it, if we start from the newly hatched chick, we assume, we know it's not quite a correct assumption, that the gut is sterile at hatch. Sure. It's not really. There are microbes present in the hatcher. There are microbes that start to colonize. The chicks are transported. They're put into a barn, whether there's fresh litter or deep litter, or previous exposure or exposure to previous flocks. There are lots of things that we can do to maybe speed that along. In Canada, we have a little bit different situation than the US. We don't use deep litter. And one of the changes that's been happening in the Canadian industry is that historically, we've always sanitized and disinfected, removed all the litter between each flock. And more recently, the rules have changed to allow a simple water wash rather than sanitation, full sanitation and disinfection. So even with removal of all the litter and a water wash, what we found was broilers placed after a water wash had less Campylobacter at the end of the production cycle than birds that were placed in a barn with a full sanitation. So that's an example of something that we can do in order to speed the development of a mature microflora. In the case of deep litter, obviously the environment microbiologically is much more complex. And so we get typically a faster transition to a mature gut microflora in those cases.
So if you had a good microflora in that house for the previous flocks, you would expect to have that established quickly.
Exactly.
I assume the converse is true, where if you had issues in that house.
Yeah, there are cases when there are challenges to gut health that can be carried over to the next flock, and then, steps need to be taken. But in general, we don't have a situation where the chick is able to peck around the environment that the mother hen is located in and develop that gut microflora. So, things like probiotics, things like exposure to a healthy microflora from previous flocks are ways that we can speed that along and provide protection by providing a more stable gut microflora more quickly.
I'm glad you brought up probiotics. There's been just a wide array of probiotics that have come onto the market as feed additives in the last decade or so. It can be kind of difficult to parse through all of the literature to make decisions on what would be the correct probiotic for a specific program. I know that's not a question that's easily answerable, but I was wondering if you have some things that you look for specifically in, product literature. What signals to you, hey, this is a well-researched product versus one that might be a little bit more of a wild, wild west situation?
Yeah, that's a great question. So there's a couple of things that I look for. One is how is it supposed to be used? How is it supposed to work? So often the microbial species that are present in probiotics will remain in the gut as long as you keep feeding the probiotic. And when you stop feeding it, they're often not competitive to remain in the gut. So persistency is one thing. Often they can be useful in terms of, very early in life, just preventing whatever comes along from colonizing. And, particularly in terms of pathogens, of course. Eventually, there will be other commensal microbes that come along that might outcompete the probiotics. So the duration, how long do you need to feed it for it to be effective? When do you feed it to be effective? Early in life or after a feed disruption or an antibiotic treatment, it might be beneficial to, again, provide a a layer of protection while the microflora reestablishes itself. Usually complex is better than simple. So if you have one bacterial species, for example, chances are it's going to be limited in the number of environmental niches or the proportion of the gut that can actually be colonized. And so if you've got a multiple strain or multiple species probiotic, there tends to be a greater coverage in the ecological niches in the gut. And then in terms of looking at research papers, there's an awful lot of, well, let me just say that proper experimental design is critical. So too often I see papers where they have a treatment where they feed antibiotics and they have a treatment where they feed probiotics, and there's no difference in performance. And so the company says our product is a replacement for antibiotics. Well, really what you need is a control with a disease challenge in there, so that when you take antibiotics out, you confirm that there is a problem there that the antibiotics are preventing. So there is some sort of, whether it's clinical or subclinical disease, but there's a loss in performance, or there's some indication that there's a disease process going on. And then if the probiotic actually restores the performance to the level of the antibiotic, or partially restores, then you can make a much more confident claim, or as a reader, you can be much more confident in the results that, yes, this might be a positive approach to the removal of antibiotics. And so that's one thing. The other thing is that disease challenges are often complex. And so if a probiotic is effective against a particular pathogen in the lab or in a small scale floor pen trial, that's no guarantee that probiotic is going to work. So it's just a reality that, you need to understand what pathogen was that probiotic shown to be effective against. And if that pathogen is not the problem in your specific operation, well, maybe that probiotic isn't the one that you should be looking at. So it's always a difficult transition to go from small scale university type research trials into full scale application. But there is a continuum. There is a progression of going from the benchtop research to the small scale pen trials to larger pen trials to testing it in commercial operations.
It can be pretty challenging to test that. It's hard to simulate a disease challenge in a controlled way in the field.
Exactly. Yeah.
Yeah, for sure. I wanted to ask you, you mentioned obviously there is some vertical transmission of different microbes, not just pathogens, but also commensal bacteria from the hen. And then there's, pathogens coming from the environment and bacteria from the environment. I wanted to ask if there's any genetic component or epigenetic component to the immune system response, and if that has been something that's influenced by selective breeding.
Yeah, so there's certainly genetic components. We have at the University of Alberta a number of random bred lines. So we have 1957 random bred broilers. They're related to the Athens Canadian random bred lines in Georgia. We also have a 1977, two 1977 lines, a male line and a female line. And all three of those lines were developed by Agriculture Canada, and we've taken them over the years. And then we have some heritage breeds, Light Sussex and Barred Plymouth Rocks and Rhode Island Reds and so on. So we've had an opportunity to look at some of the differences in immune function among those different strains. And certainly as we're selecting for growth rate, typically we're selecting against, and I need to be careful about how I say this, we're selecting against the systemic effects of inflammation. So I had an undergrad student do a project, and unfortunately this never got published, but what we found was that the modern broilers have a lower systemic inflammatory response. So if we look at mediators of inflammation, they tend to produce less of the signals, the pro-inflammatory signals like interleukin-1, than the random bred broiler lines. Now, what was interesting is when we looked at the immune cells, T cells, for example, the T cells of the modern broilers were much more sensitive to a given level of interleukin-1 than the random bred broilers. So, I want to be careful and not say that, the modern birds have an impaired immune system, but I think that selection for growth rate has brought along unintended changes in the immune system. And I think that the modern birds do the same kinds of things, but they do them more efficiently from a growth standpoint. So to the same challenge, they're less affected by fever and reduced appetite and inefficiencies in nutrient utilization that come along with a strong inflammatory challenge.
That makes a lot of sense.
I've always wanted to go back and redo that with the birds that we have now.
Yeah, that would be a very interesting project. I could see how if you're selecting for example, feed efficiency, I mean, you probably as part of that are selecting for the bird to not expend as much energy in an immune response.
Exactly. And, I think with the change towards removal of growth promoting antibiotics, it's exposed maybe some of the limitations that might be present. So again, it's important to develop new strategies and new techniques. I should point out that I think the primary breeders have done a really good job of including health in their selection programs, and both in terms of metabolic health and also immunological health. And I know in speaking with the geneticists for years and years and years, they have done their selection in an antibiotic-free environment. So they've been taking that into account. And so I think that's, we still see increases in growth rate, increases in efficiency, even with the removal of antibiotics. So I think the geneticists have done a good job.
Mm-hmm. Absolutely. They've been working very hard on trying to select on so many things, and it's hard to select on everything.
Exactly.
So it just takes time. Yeah. Before we kind of change gears and talk a little bit about layer nutrition, I wanted to see if there's anything else that you wanted to talk about in the immune and gastrointestinal health arena.
Just in working a fair bit with replacements for antibiotics or alternative strategies, it's really become apparent that there isn't a single magic bullet that we take antibiotics out and we put this other thing in and we can just pick up where we left off. So it becomes more challenging, I think, in terms of understanding just what the problems are in a particular location or within a particular company. And even it's not even within a particular company, it's a particular complex within that particular company has a different set of needs than a different complex even in the same company. So talking to some colleagues who are also producers, it takes some trial and error to understand what are the challenges that they typically face, what are the products that are most likely to be effective against those specific challenges. And you have to do that on a company by company or location by location basis. So it's more challenging, but it's certainly doable.
Mm-hmm. I can see that being very challenging, especially when, logistically things have become so centralized, especially on the feed side. As much as we would like to precision feeds some small subpopulations of a complex who are having an issue, it's just very difficult to do. Maybe we'll move back towards more smaller feed mills to do that. I don't know. Or on-farm feed blending, something. But yeah, very tricky. So on the subject of layers, I have to confess that I wouldn't consider myself to be a layer nutritionist by any means. I'm definitely much more broiler biased in my career and training. So you'll have to forgive me. I wanted to just ask, since you are an expert in the area, what are the hot topics in layer nutrition right now?
So I think the biggest thing is the transition towards long layer cycles. So rather than a forced molt and a second production cycle, the geneticists have done a really good job of selecting for health, for longevity, for persistency, And probably most importantly, they've selected for a much more or much less rapid increase in egg size as hens get older. So one of the limiting factors has always been shell quality, and as the eggs get bigger and bigger and bigger, as the hens get older and older, shell quality tends to suffer. So by reducing the rate of increase in egg size, they've been able to breed a bird that has good quality shell for long production cycles. So, up to 100, I've seen 120 weeks. We just finished an experiment earlier in the month where we had a flock of laying hens go to 106 weeks of age. So for our research program, that's a long time to have a flock of laying hens. So, but I think it's important because that's what industry is doing, and so we want to be able to translate our results into industry as seamlessly as possible.
How does that longer laying cycle impact the nutrition of the bird?
Yeah, so as hens get older, typically their efficiency of calcium and phosphorus metabolism decreases. After peak production, they have a fairly fixed ability to deposit shell onto the eggshell. So as the eggs get bigger and bigger, with a fixed amount of shell, the shells have to get thinner. So that's why that slowing the rate of increase in egg size is so important. So one of the challenges over the years has been trying to figure out, optimize calcium and phosphorus nutrition as the birds get older. I had a master's student who just defended, and we're working on a meta-analysis of calcium requirements. And probably to my surprise, we found that there is, I expected to see that as we went higher and higher in calcium in laying hen diets, we might start to see some negative effects because of interactions with phosphorus, interference with the activity of phytase. We didn't see that to the extent that I would have expected. So the hens seem to be pretty resilient as long as it's above the requirement. The hens seem to be pretty resilient in terms of egg production, in terms of bone health, to increasing levels of calcium. And that, like I said, that surprised me. What we're finding, and I think is becoming clearer, is that phosphorus is probably a major player in terms of shell quality. And so rather than birds being deficient in phosphorus, I think it's becoming clear that typical layer diets are too high in phosphorus. And so we just published a paper with Marcus Rode-Hutskord from Hohenheim University, looking at, this is carrying on from a meta-analysis that he and a colleague had done, where they found, based on the literature, laying hens probably don't require any more than 0.22% available phosphorus. And typical diets are above 0.4. So that's a lot of phosphorus going into diets. And then when phytase is used, that liberates even more phosphorus in the gut. And what we tend to see is the formation of calcium phosphorus precipitates, and we actually interfere with calcium absorption. So I think there's a lot of opportunity for the layer industry to reduce the amount of phosphorus in the diet, reduce diet cost by reducing inorganic phosphate supplementation. And if we go even lower, then we have the opportunity for phytase to become effective. So I think when we have high phosphorus diets, adding phytase is costly and it's counterproductive.
Do you think you can get away with just phytase?
Well, we'll find out. So the experiment that we just finished up, from 80 to 106 weeks of age, we took out all of the inorganic phosphate. So the birds are still laying well. We haven't analyzed shell quality yet, or we haven't run the statistics, and we still have to look at bone quality. But in terms of production from 80 to 106 weeks of age with no additional inorganic phosphate, the production was just fine. So what we think is possible is when we use phytase at the appropriate level, we can take the inorganic phosphate out throughout the egg production cycle. So we're working on a grant right now to investigate that all the way from day-old pullets up to 100 weeks of age.
Wow. I mean, that's great research. And by the way, very applied. So you're doing both. You're doing both ends of the spectrum.
Yeah. And so the phosphorus is, phosphates are limiting or limited the supply, and they're expensive. So whatever we can do to reduce those diet costs by eliminating an expensive ingredient that's really not needed, that's going to be good for producers.
Not to mention the sustainability impact that, phosphorus in manure is quite a troubling problem sometimes.
Yeah, in a lot of regions. Yeah.
Mm-hmm. Before we wrap up, is there anything else about layers that you'd like to talk about?
Well, one of the things that I talk about when I'm speaking to industry is I spend a lot of time talking about pullet nutrition. So when those birds are kept for longer and longer periods of time, they're laying more and more eggs per hen. The pullet nutrition just becomes critical. So it's really important, whether you're growing your own pullets or buying in point-of-lay pullets, that they have been managed properly, they've been fed properly. And I always tell producers to start with the end in mind. So with that day-old pullet, you need to have a plan for how you're going to get the bird to the point where she's ready to lay, and not just ready to lay, but ready to sustain 80 to 100 weeks of egg production.
That's good advice. Very helpful. Well, to wrap up the show, we typically ask the same 3 questions of all of our guests, and these are relating to resources that you would recommend to our audience. The first of which is, can you recommend a favorite book or resource related to poultry nutrition?
Well, I think the Poultry Science Association website, and so the directory, that's where if I'm looking to contact somebody, that's often the place I look for the journals, for the meetings that Poultry Science puts on, and just over the years at the Poultry Science Association has been a great resource, and I use their website a lot to make the connections and read the papers, and so that's my recommendation.
Absolutely, that's a great recommendation. How about a book or resource outside of your field? So this could be anything that you find personally enriching.
So I'm a bird watcher. There is a website called eBird, and it's run by the Cornell Lab of Ornithology. And so this allows me to track and record the birds that I've seen. It's a great resource. So when I go to a new place and to give a talk or to do some consulting, I often try and spend an extra day or two and get some bird watching in. So it helps me to scout out locations. And it's just really interesting from being able to see what I might expect to see and hope to see at different locations when I go bird watching.
Very cool. I've recently gotten into bird watching as well, and it's a really fulfilling pastime.
Yeah, and I always say that even if it's a bad day of bird watching, it's still probably a good hike.
Yeah. You still get to see some birds. They may not be the birds that you came there to see, but usually you see something. Very good. And then lastly, a more philosophical question. If you think about what makes a successful person, could you tell us about the qualities or characteristics that you think lead to success? And this could be in poultry science, in industry, in academia, or just in life in general.
I think curiosity is one of the big ones that's underappreciated. And so over the years, I've had a lot of grad students, and I've enjoyed working with each of them. But I found that, often the academic superstars, if they're not curious, are the ones that end up struggling the most, even though they're, probably the smartest people. But that curiosity and that willingness to ask questions and to try and look at things from different angles and try and solve a puzzle. I think that's something that over the years I've learned is really important. And one of the things that I talk to prospective grad students about is, what are you curious about? Or would you describe yourself as a curious person? Or, how do you approach a new problem? And I think that curiosity and that desire to learn, to advance without being able to necessarily go to a book and find the answer. That's a critical characteristic, I think, in having a successful career in science.
It's a wonderful response. I definitely agree. I think to be curious, you have to be willing to take the risk of looking a little foolish almost to ask the questions without worrying what people will think about you asking the question.
Yeah, that's a great way to put it.
Yeah, and that is something that a lot of people struggle with, including myself sometimes. So, well, thank you so much for your time and your insight and all the wonderful research that you do. We wish you the best of luck in your future consulting role. Hope to see you out there among the birds.
Well, thanks for inviting me on the program.
Yeah, thank you so much for being here. I hope you have a great rest of your week.
Thank you, you too.